Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine knowing years in advance if Parkinson's disease was maybe on your horizon long before the first tremor even, thought about appearing. Sounds like something out of a science fiction movie, right?
It really does.
Well, get ready, because we're diving deep into a fascinating scientific development that's making this possibility, well, a whole lot closer to reality.
Yeah. Today we're taking a close look at a brand new ribonucleic acid-based blood test. It's designed to detect Parkinson's disease.
Okay.
Our mission, basically, is to understand how this test works, like fundamentally, explore the impact it could have, which is potentially huge, and also share what the researchers themselves are saying.
And we've got the source material, right?
We do. We've got a health news piece that really highlights the early detection side of things, and then the detailed scientific paper itself. It's called Pre-Symptomatic Parkinson's Disease Blood Test, Quantifying Repetitive Sequence Motifs in Transfer Ribonucleic Acid Fragments. Quite a mouthful.
Yeah, a bit technical, but we'll break it down. And for you, the listener, why tune in? Well, if you're someone who likes staying ahead on health and science, you enjoy those aha moments with complex stuff.
Or just want the inside scoop without the jargon.
Exactly. Then this deep dive is for you. We're going to pull out the core insights, explain the science simply, and talk about why this is generating so much excitement. So let's jump in.
Okay, so let's start with the problem they're trying to solve. Right now, Parkinson's disease, it's usually diagnosed pretty late in the game.
After symptoms are already obvious.
Precisely. After significant damage has already occurred to the nerve cells, usually when motor symptoms like tremors become noticeable.
Right, but we know the disease doesn't just, pop up overnight with the tremors.
Yeah.
There are earlier signs.
Absolutely. Things like losing your sense of smell or certain sleep disorders like rapid eye movement, sleep behavior disorder. These can show up years before the motor stuff.
Years.
Yeah, years. So this suggests there's this long pre-symptomatic window. And if we had a reliable way to spot the disease then, well, that could open up whole new avenues for intervention.
So that was the goal then, to find a way into that window.
That was the main objective, to develop a simple, cost-effective blood test that could catch Parkinson's in those very early pre-symptomatic stages. The researchers actually drew a parallel to cancer diagnosis, decades ago. Back then, detection was often late, limiting treatment options.
Ah, okay. So they're hoping for a similar shift for Parkinson's. Earlier detection, better outcomes.
That's the hope. A blood test for a brain disease, identifying it potentially years early. It's a pretty ambitious goal.
Definitely ambitious. So what was their novel approach here? How does this test actually work?
Well, what's really interesting is it doesn't look directly at the brain. Instead, it focuses on quantifying specific ribonucleic acid fragments floating around in the blood.
Ribonucleic acid fragments.
Yeah.
Okay, let's unpack that. What kind of fragments are we talking about?
They zeroed in on two key types that seem to act as biomarkers, indicators. First, they found elevated levels of certain transfer ribonucleic acid fragments, or tRNA fragments, that carry a specific repeating sequence motif.
A sequence motif, like a pattern.
Exactly, like a pattern. Think of it as a kind of molecular barcode. The sequence is RGTTCRA, which stands for either adenine or guanine, then guanine, thymine, cytosine, and then adenine or guanine again.
Okay, that specific sequence.
Right. And these particular fragments were pretty long, over 30 bases, meaning they're substantial. And interestingly, they seem to come from lots of different transfer ribonucleic acid genes found in the cell's nucleus.
So biomarker 1, more of these long tRNA fragments with the RGTTCRA barcode. Got it. What was the second biomarker?
The second one was the opposite, actually. They found reduced levels of tRNA fragments that originate from the mitochondria.
The mitochondria, those are the cell's powerhouses, right?
That's them. And this decline in mitochondrial tRNA fragments is thought to reflect the deterioration of the mitochondria as Parkinson's progresses. These were mostly the long fragments found at one specific end, the 3' end.
Okay, so fewer the mitochondrial ones. We have an increase in the RGTTCRA type and a decrease in the mitochondrial type. How do they combine these for the test?
Well, the key wasn't just looking at one or the other in isolation. It was the ratio between them.
The ratio?
Yeah, the balance. Measuring the ratio between those elevated RGTTCRA barcode fragments and the reduced mitochondrial fragments. That specific ratio seemed to be what could effectively distinguish people in the very early pre-symptomatic stages from healthy individuals.
So it's like the cell's distress signals more of one type, less of another, and the ratio captures that overall distress level.
That's a good way to put it, yeah, even before the obvious physical symptoms show up.
Okay, that makes sense. So what evidence did they gather to back this up? It sounds promising, but they must have tested it thoroughly.
Oh, absolutely. They built a strong case. First, they looked at people carrying specific gene mutations linked to a higher Parkinson's risk, like LRRK2, GBA, or SNCA mutation.
Right, the genetic risk factors.
And they found that these individuals, even if they had no symptoms yet, had higher blood ratios, that RGTTCRA to mitochondrial tRNA fragment ratio, compared to other people who had the same mutations but didn't show signs later.
Wow. So the test picked up something even before the clinical signs appeared in those high-risk individuals.
Exactly. Suggests it's tapping into the very early disease process.
That's pretty compelling. What else?
They also looked at patients already getting treatment, specifically deep brain stimulation, which helps manage motor symptoms.
Great.
And interestingly, after the treatment, the levels of those RGTTCRA barcode fragments in their blood actually decreased.
Huh. So the treatment that helps symptoms also affects this biomarker. That suggests a connection.
It definitely suggests a link, yeah. Maybe to the mechanisms underlying the symptoms themselves.
Fascinating. Any lab work?
They did some work in lab-grown nerve cells, neuroblastoma cells. They found that these RGTTCRA fragments seem like they could potentially interfere with how the cells make proteins.
How so?
Well, they appeared to have parts that could bind to the cell's protein-building machinery, maybe gumming up the works, which hints at a possible way these fragments might actually contribute to the disease at a cellular level.
So they might not just be passive markers, but actually involved in the pathology.
That's the potential implication, yes. And it didn't stop there. They also analyzed postmortem brain tissue.
From the affected area.
Yes, specifically the substantia nigra, the part of the brain hit hardest by Parkinson's. And they found the same pattern: higher levels of the RGTTCRA fragments and lower levels of the mitochondrial ones.
Just like in the blood.
Just like in the blood. And importantly, these changes in the brain tissue correlated with higher Lewy body scores. Those are the microscopic hallmarks of Parkinson's pathology. So the blood signals seem to mirror the brain changes.
Okay, that strengthens the link significantly.
There was one slightly odd finding, though. While mitochondrial tRNA fragment levels were down in the cerebrospinal fluid and substantia nigra,
Yeah.
in postmortem blood samples from advanced Parkinson's patients, the mitochondrial fragments were actually elevated.
Elevated, why would that be?
The researchers think it might be an artifact of death, essentially, that mitochondria might break down more readily in blood after death, releasing more fragments. It just highlights, the complexities you encounter when studying biomarkers. Context matters.
Definitely. Lots of evidence, but also those complexities. So how did they turn all this into a practical, usable test?
Right, good question. To make it standardized and easy to compare results, they developed a quantitative polymerase chain reaction assay, qPCR.
PCR, okay.
Yeah.
That amplifies specific genetic material.
Exactly. They designed two specific short DNA sequences called primers. Think of them as like highly specific search probes. One primer is designed to find and bind to lots of those RGTTCRA barcode fragments targeting a specific sequence. Guanine, thymine, stamine, cytosine, thymine, guanine, thymine, cytosine, adenine.
Okay, a specific target for the first biomarker.
And the other primer targets the mitochondrial tRNA fragments, looking for the sequence thymine, adenine, cytosine, thymine, adenine, cytosine, adenine, guanine, cytosine, adenine, cytosine, thymine, adenine.
Got it. So the qPCR uses these primers to count how many of each specific fragment type are in the blood sample.
Precisely. It quantifies them. And the ratio of the counts detected by these two primers gives you what they called the transfer ribonucleic acid fragment score, or tRNA fragment score.
The tRNA fragment score, okay.
Yeah.
And how well did this score actually perform? Did it work?
This is where it gets really promising. They used machine learning algorithms to see how well the score could distinguish between people in that pre-symptomatic phase and healthy controls.
Yeah.
It actually outperformed the standard clinical assessments used today, like the Unified Parkinson's Disease Rating Scale motor scores or the Hone and Yar staging.
Better than the clinical scores. By how much?
Statistically, the measure they used, area under the curve, or AUC, was 0.86 for the tRNA fragment score, compared to 0.73 for the clinical measures. An AUC of 1.0 would be perfect, so 0.86 is pretty good. especially compared to 0.73.
Wow, okay. That suggests it's potentially more sensitive at picking up these very early stages.
That's the indication, yeah. And it seemed pretty robust across different groups, too.
Meaning?
Meaning the score could differentiate pre-symptomatic folks from controls across various ethnicities and genetic backgrounds. However, and this is important,
Okay.
they noted that Black individuals in the control group and the pre-symptomatic group had quite similar tRNA fragment scores. This mirrored what they saw with the clinical scores in that group, too.
Ah. So it wasn't as clear-cut for Black participants in this study.
Right. Which really underscores the need for more research in diverse populations. We need to make sure it works well for everyone.
Absolutely. Critical point.
Right.
Did they validate these findings in like a completely separate group?
They did. They tested it on a separate holdout group of pre-symptomatic patients and controls, and the test's effectiveness held up.
Good.
And even more tellingly, the tRNA fragment score showed an even bigger difference between controls and those pre-symptomatic individuals who actually went on to get a formal Parkinson's diagnosis later compared to how the clinical scores performed.
So it seems particularly good at identifying those who are truly on the path to developing the disease.
It appears so. And just to cross the T's, they also ran a manual version of the qPCR test on fresh blood samples. They compared diagnosed Parkinson's patients, trauma patients, and healthy controls. Same result. The Parkinson's patients had significantly higher RGTTCRA to mitochondrial tRNA ratios than the other groups. They saw similar patterns in postmortem brain tissue, too, using a slightly tweaked primer for the brain.
Okay, so a lot of converging evidence supporting this tRNA fragment score.
Ah.
Let's pivot now to the implications. What could this actually mean for Parkinson's disease?
Well, the biggest implication really is early detection.
Getting ahead of the curve.
Exactly. Having a non-invasive blood test that could spot Parkinson's years before the major motor symptoms hit, that could be completely transformative.
How so?
It opens up that critical window for early intervention, maybe even before irreversible nerve cell damage happens. And think about developing new drugs.
Clinical trials.
Right. If you can identify people who are definitely in the early stages, you can test therapies designed to slow or stop the disease much more effectively and much faster. It can massively accelerate the development of disease-modifying treatments.
That'd be huge. Speeding up drug discovery. Any other impacts?
Well, it could also make clinical trials themselves more efficient. You'd have a clearer group of participants who are likely to progress, and thinking longer term, maybe personalized diagnostics.
What do you mean?
Like comparing an individual's tRNA fragment score not just to healthy controls, but to Parkinson's patients with similar backgrounds, age, sex, genetics. It might give a more tailored risk assessment or prediction.
A more precise picture for each person. That's the dream of personalized medicine.
It is. And remember that finding about deep brain stimulation, how the RGTTCRA levels dropped after treatment.
Yeah.
That hints that this test might also be useful for monitoring disease progression or seeing if a treatment is actually working biologically in a patient over time.
So diagnosis, trial efficiency, personalization, monitoring, lots of potential angles.
Definitely. But, as with all research, we have to talk about the limitations, too.
Crucial. No test is perfect, especially early on. What are the caveats here?
Well, we already touched on one big one, demographic specificity. The study relied heavily on certain groups, and the finding with the Black cohort shows we absolutely need more research in diverse populations to ensure it's reliable and fair for everyone.
Non-negotiable, really. What else?
The study design was mostly cross-sectional, meaning it was like taking a snapshot at one point in time.
Not tracking people over years.
Exactly. To really understand how these biomarkers change as the disease progresses or how they respond to treatment long-term, you need longitudinal studies following the same people over many years.
That makes sense for a progressive disease like Parkinson's.
And finally, the researchers themselves are very clear. These findings need validation in larger independent groups. Their initial validation was good, but confirming it in bigger, more diverse cohorts is the essential next step before anyone thinks about using this routinely.
Right. So promising, but more work needed. Responsible science.
Okay, let's bring it back to the researchers. What were their main takeaways based on the paper? They seemed pretty convinced that these tRNA fragments, especially the ones with those specific sequence motifs like RGTTCRA, are really promising biomarkers. They even use the term patient-specific fingerprints.
Fingerprints. I like that. Very individual.
And they emphasize the consistency they saw finding the link between these fragments and Parkinson's across different disease stages and in different fluids, brain, cerebrospinal fluid, blood. That consistency suggests a strong fundamental link to the disease.
And the origin of some fragments seemed important too. Something about dopamine?
Yeah, they noted that some of these RGTTCRA fragments come from tRNAs involved in making dopamine and also in antioxidant processes, both really relevant to Parkinson's pathology. This hints that the fragments might have a functional role, not just be bystanders.
Right, disrupting key processes perhaps.
Possibly, and they were definitely encouraged by the test's ability to distinguish those pre-symptomatic patients, potentially better than current clinical scales. That's key for early diagnosis.
And the practicality of it being a blood test.
Huge advantage. They highlighted that developing this relatively simple qPCR assay makes it way more accessible and less invasive than, say, needing a spinal tap for cerebrospinal fluid.
Makes sense. Easier, cheaper, less risky.
And finally, they pointed to that connection between the fragment levels changing after deep brain stimulation and the lab findings about protein production, suggesting these RNA bits might be tied into the actual mechanisms of the disease and how treatments work.
Okay. So summing up, this research really gives us a compelling glimpse into a potential future for Parkinson's diagnosis, doesn't it?
It really does. The possibility of detecting it years early, that could just revolutionize everything. Research, treatment, patient care.
Yeah. And it makes you wonder, right, what are those next steps? Validating it properly across diverse groups seems paramount.
Absolutely. And what about new therapies? Could we actually target these specific RNA fragments somehow? That's a whole other exciting avenue.
It really is. So for everyone listening, we'd encourage you to maybe dig a bit deeper into neurodegenerative research. Think about what early detection could truly mean for people affected by Parkinson's.
Definitely. Keep an eye on this field. It feels like we've just opened a really significant new chapter in understanding this condition.
A hopeful chapter for sure.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.